Good morning everybody, I choose this opening for the topic I'll talk about today, the third dimension and how can we connect it to the infinitely small, with 3D microscopy techniques. The third dimension refers to depth, the spatial axis that alongside height and width, allowing objects to occupy a volume.
Imagine a vast plain, a world of only 2 dimensions, on which triangles, squares, pentagons, and other figures live and move freely about.
Configuration makes the man. Calculate.
Get to your squaricle! Now!
You're only a square. Thanks, brother.
They know nothing of our 3-dimensional world.
Such a notion is, of course, absurd and furthermore, illegal.
But that's all about to change.
Where did you come from?
I come from space, the 3rd dimension.
This was the trailer of a short movie based on the romance Flatland, written by Edwin A. Abbott. As you have seen, Flatland is a world that exists only on the 2-dimensional plane, where its inhabitants, literal geometrical shapes, live in a highly structured society organized into classes based on the number of sides of a figure. The narrator and protagonist of the romance is a square who reads from prison about his encountering with a sphere that revealed to him what is inconceivable for Flatland's inhabitants, the 3rd dimension. At the end, for speculating about even more dimensions, the square ends up imprisoned by other citizens. Flatland offers a powerful metaphor for how perception limits understanding. Just as the square cannot imagine a 3rd dimension until he experiences it, similarly, 3D microscopy reveals structures in biology that 2D imaging can't capture, adding depth to our view of life at the microscopic scale. So, like the square, scientists break through dimensional barriers to uncover hidden layers of reality. I hope this introduction has made you curious about using microscopy to obtain 3D images. Now I'll start with a brief overview on microscopy and microscope components to finally tell you more about 2 types of 3D microscopy techniques, and better explain you the one that we use in my research laboratory. Talking about how can we see, the cornea acts as a lens, projecting an image onto the retina. The iris adjusts its diameter to control light entry, and the crystalline lens changes shape, allowing the eye to focus on objects from about 20 centimeters to infinity. But the details we want to observe may have a diameter of only 1 out of 100 of a millimeter, and so, as told before, it is not possible for us to get closer to the object than approximately 20 centimeters. As a consequence, the visual angle is reduced and we are no longer able to recognize any details. So what is the solution to see more details? We can use a magnifying glass between the eye and the object to make things appear larger, but this effect is limited. It is not possible to obtain a magnification greater than 8 or 10 times. So if you want to see more, you need to use a compound microscope. A standard light microscope is made of the light source with a diaphragm to change the amount of light, and condenser lenses that are optical components located below the stage to focus and concentrate light into the specimen, providing a uniform illumination and enhancing clarity and contrast of the image. The stage, where we place the specimen, that can be moved to see different parts of the sample. Objective lenses located near the specimen, normally in a number of 3 or 4, in a revolver, to choose the best one for your specimen. In fact, they come in different powers, for example 10x, 40x, 100x, and produce a real inverted image of the sample. And at the end, ocular lenses, also called eyepieces. They are located at the top of the microscope where you place your eyes, and they further magnify the image produced by the objective lens, typically by 10x, and present a final virtual image to the observer. And the microscope can be binocular or trinocular due to them. Total magnification is calculated with the multiplication between ocular and objective magnification. A first 3D microscopy technique is the stereo microscope one. A stereo microscope uses two separate objectives and two independent optical paths, allowing each eye to see the object from a slightly different angle. It relies on reflected light rather than transmitted light, making it ideal for observing the surface of opaque specimens. This design provides true two-dimensional images with a sense of depth, and the typical magnification ranges from 10x to 50x, offering clear detailed views of larger samples. Due to its low power range and surface view capability, it's most often used in entomology and parasitology. A second technique I'd like to present you is the 3D fluorescent microscopy. Fluorescence describes how certain substances absorb light and almost immediately re-emit it. A fluorochrome is a molecule that absorbs light, so its electrons jump to a higher energy state and then realize this energy as light of a longer wavelength. This difference is called the Stokes shift. In fluorescence microscopy, we use this principle with visible light, which ranges from about 400 to 700 nanometers. We use this to study structures at the microscopic level. The graph reported represents the wavelengths used in fluorescence microscopy to excitate the sample, obtaining an emission of a certain color in the spectrum. A fluorescence microscope is an optical microscope that uses fluorescence instead of, or in addition to, reflection and absorption to generate an image and study properties of organic or inorganic substances. It can have a simple setup like a wide-field epifluorescence microscope or a more complicated design such as a confocal microscope I'll talk about later. The typical optical path for an epifluorescent microscope is the following one. The light generated by a light source reaches the fluorescent tube and is filtered by an excitation filter that selects the excitation light. The short wavelength excitation light is reflected by a reflecting mirror into the specimen through the objective. The emission produced, which has a wavelength greater than the excitation light, is collected by the objective and transmitted through the dichroic beam splitter, reaching the lens of the tube and the eyepieces. If some light is reflected by the sample, to avoid it to reach the detector generating a fluorescent background, an emission filter captures it. Let's consider now our biological samples. They are not flat, 2D, they have a three-dimensional shape, so we have to consider the z-axis of depth. The resolution of the z-axis, axial resolution, is lower, the limit is 500 nanometers, while the lateral resolution of x and y-axis has a limit of 200 nanometers. Including this fact that affects the quality of our images, we need an improvement in microscopy techniques that permit better resolution of 3D sample images. In fact, the main problem is that when you excite a sample containing fluorophores at different planes, you cannot excite all of them equally. The light transmitted passes through the sample, and the fluorescence emitted by fluorochromes in the focal plane appears sharp, while the light from out-of-focus planes is more blurred. This diffuse light from out-of-focus areas reduces the quality and the contrast of the final image. The solution to this problem is taking optical sections, an approach to virtually section our sample, because if we are able to detect the light deriving from only a specific plane, blocking the light deriving from other planes, we obtain a clear, sharp fluorescence image, not disturbed by out-of-focus light. How is this possible? There are different optical sectioning methods. The first is avoid out-of-focus light, the excitation strategy, used in multiphoton microscopy and total internal reflection microscopy. The second are confocal methods based on detection strategy, so all fluorescence molecules are excited, but we detect only the ones we want to. And the third is removing afterwards the autofocus light. Confocal microscopy is one of the most widely used optical sectioning techniques today, offering a powerful balance of image clarity, depth resolution, and ease of use in biological and biomedical research. The main difference between wide-field classical fluorescence microscope and confocal microscope is that in the second one, only the light deriving from focal plane arrives to the detector, thanks to a detector pinhole aperture. So, the confocal image readily resolves small structures and reduces out-of-plane fluorescence. The confocal laser scanning microscope scans the sample sequentially, point by point and line by line, and assembles the pixel information to one image. By moving the focus plane, in fact, single images, the optical slices, can be put together to build up a three-dimensional stack that can be digitally processed afterwards. So with this technique, optical slices of the specimen are imaged with high contrast and high resolution in all X, Y, and Z axes. This video showcases real-time cell division seen through a confocal laser scanning microscope, specifically highlighting how cellular structures like chromosomes and microtubes are visualized using fluorescence. It clearly demonstrates how the optical sectioning capabilities of a confocal microscope enable us to see detailed 3D-like organization within live tissue samples. I decided to end this presentation showing you how a confocal fluorescent microscope works in reality. So this is Leica SP8 microscope provided in the laboratory where I am attending my internship. The Leica SP8 is indeed a lightning confocal laser scanning fluorescence microscope. It means it has a lightning mode, an integrated automatic deconvolution technology that reveals finer structural details. And as a confocal microscope, it has a pinhole in front of the detector that blocks out-of-focus light. It uses lasers as light sources to excite fluorophores in the sample, and it has a scanning system that scans the laser point by point over the specimen. So it can create high-resolution, three-dimensional images by stacking multiple optical slices. This is the room dedicated to the Leica SP8 microscope. It is important to keep it dark to allow the detection of even very weak fluorescence signals. Leica SP8 is composed by the microscope, the screens, and the control panel. This is the microscope structure. Eyepieces, fine and coarse focusing, adjusting knob to move the stage in X, Y, and Z axes. The stage where is placed the slice with the sample. The body of the microscope I was talking about before. The objectives placed down the sample as an inverted microscope. Let's see a slice analysis at the microscope. First, we clean the slice, the part not containing the sample, with ethanol to remove montant residues. Then we add a drop of oil in order to use an immersion objective to obtain brighter, clearer and more detailed images thanks to the creation of higher numerical aperture, and so better resolution by reducing the light loss and refraction at glass sample interface. Now we place the sample upside down, cause the objective is placed down the stage as an inverted microscope. Before using fluorescence lasers, we locate the area of interest using bright field in order to find where our cells are. This is because if the specimen is not yet to be observed, we avoid the excitation light to prevent light from fading the specimen. Now to focus the image, we use the DAPI signal, which stains cell nuclei blue. The signal is clear and easily recognizable, making it ideal for precise focusing. And this is what the observer can see through the oculars. Now we can concentrate on the signals we are searching for in this sample. In our case, a red range signal, so the microscope uses a green laser to excite it, because as we said before, the excitation light must have a shorter wavelength than the emitted fluorescence will obtain. We can then move the visualization to the screens. Obviously, all the machinery was set before its use, specifically for the type of sample we want to analyze. However, the most important step talking about 3D microscopy techniques is now to set the zeta stack. It means to define the limits between which the microscope will capture a series of images, each one at different focal planes along the zeta axis. And this was the result of the overlap of each optical section, where we wanted to highlight focal adhesions of the cells. Thank you for your attention.